Good afternoon, everybody. I hope I'm audible as well as visible to you all. Please let me know if my presentation is visible to you. Thank you, Devangana. Just join through my mobile so I can. Yes, Kritika will be looking into it. Yeah. So regarding the calculator issue, just I had mentioned last week, you will have to do it manually. Uh, it is possible to do. I mean, we have all been doing this during our school 11th, 12th, or 10th mathematics. So it's not an impossible thing, and it's of a single degree polynomials only. So I am planning on doing one of the problems that we had already solved last week, or we can take up one more problem as well from the question, I mean, week 10. And let's do it manually ourselves, and so you are confident that it is doable and you can attempt during the exam. So, I mean, calculator makes life easy, but it's not necessarily the only way, or you are not absolutely dependent on it to solve that question. So, we'll look into it at the end of the class, probably. Yeah, we will do it, Kritika. Anything else? So, shall we start the class? Today's session, I think the assignment is uh, comparatively really simple. So, should all be able to participate and answer. And Kritika, I also got your mail. I will look into the issue and solve it. Okay. So I welcome you all to the 12th live interaction session for the course Bioinformatics, Algorithms and Applications. And I believe this will be the last session for this course during this NPTEL run. Let's get into the session. Yes, Devangana, the virtual calculator will be there, but it will be basic. I did not check the link, uh, but based on my experience while taking up online exams or such competitive exams, I mean, you will generally not have an equation tab or option to solve multiple equations by regression. Okay, so that is something that we have in our scientific calculators. And since I had used my calculator while solving the problems during the live session, maybe it is assumed that you need a calculator to solve those. I mean, we have done in 10th or 11th definitely during our math exams and papers, question papers, answer sheets to solve multiple equations and of greater difficulty. So this should be a cakewalk. We'll do anyway to refresh ourselves, you know, look back into our memories. Okay. So this week the practice questions are on applications of bioinformatics parts 1 and 2, actually majorly on that, and overview parts 1 and 2 of all the topics that we've been discussing, and also covers demo of Vika. So let's start with the first question. Choose the correct statement. Option A, SVM is used for solving classification problems. Option B, SVM is used for solving clustering problems. C, SMV is a supervised learning algorithm. And D, none of the above. Thank you, Shreya. Uh, think about the question again, and I don't have any typos. If you need a clue, both Devangana and Shreya. And you might have noticed my class is not on Friday this week, and Saturday I had to swap with one of my colleagues who wanted Friday's class. But I'm still happy to see some of the familiar names I see on Friday. Thank you, Shreya. Thank you, Kritika. All right, 4 votes for A. So now we are clear, right? This is just a, I don't know how the question was framed. Was it to see if we are awake in reading with all our attention? So that removes C from the option and also none of the above. So amongst A and B, we know that SVM is used for solving classification problems and not clustering problems, right? Because it identifies support vector machine is a method to identify a hyperplane which separates two classes in space, in an n-dimensional space, and so it's definitely for classification and not for clustering. So you have option A as the right answer. Question 2. In Veka user interface, which tab is used for loading the dataset and applying filters? Is it pre-process or classify or associate or cluster? Okay, 4 votes for A. That's it. All right. I mean, pre-process is of course the tab under which you load the data set, sorry, and also apply the filters, not classify or associate or cluster. Option A is right. Question 3. Uses of Veka are A. It is free and open source and provides a simple graphical user interface. B. It can directly provide the AOC, which I believe is area under the curve. C. It contains a collection of state of the art machine learning algorithms and data pre processing tools. So, which one is it? One that says only A and B, or two which says only A and C, three that says A, B, and C, and four none of the above. Okay, Debanganas is 2. Also Purnima and Kritika. I believe Kritika meant B for 2. Okay. So yes, Vika is free and open source. Provide the simple GUI, which is true. And B, it can provide AUC. That's wrong. C, it contains a collection of state-of-the-art machine learning algorithms and data pre-processing tools. Again, that's true, right? So it leaves 2 as the right option. Question 4. Which of the following is not a metric to evaluate the performance of a method? Is it A, precision, B, recall, C, association rules, and D, accuracy? Thank you, Shreya. Thank you, Malini. Let's wait one more minute. Great. So no takers for B or A. All right, you guys are right. So of course we use precision and accuracy for as a metric to evaluate performance of a method, right? So in case some of you haven't heard of recall, is also a classification metric used to measure the method's performance, represented by total number of true positives divided by total number of actual positives, which is similar to sensitivity, right? So recall is described as a way of remembering or uh, making insights based on the past. How well the model recalls something that it has already experienced or done before. So it has association rules, which is not a metric to evaluate performance of the method. Question 5. Consider a confusion matrix in which, Purnima, this is for previous question, I believe, right? Or this one. Okay, consider a confusion matrix in which true positive is 70, false positive is 8, true negative is 38, and false negative is 5. Which of the following statement is correct? A, that accuracy is 81%, B, misclassification rate is 19%, C sensitivity is 93.33% and D specificity is 13.67%. Take a couple of minutes. Tell me what the answer is. Thank you, Devangana. I'm still waiting for answers, so don't think because I began writing, I started solving. Thank you, Kritika. For others who are yet to solve, accuracy is given by true positives plus true negatives, right? Divided by all of these true positive, true negative, false positive, false negative. So, what does that give you? TP plus TN will be 108 divided by 121. So, this is definitely not going to be just 89%, right? So, that's gone. People who are still solving, you have B, C, and D to guess from. Let's first calculate what specificity is, which is true negatives divided by true negatives plus false positives, right? So true negative is 38 divided by true negative plus false positive is 8. 38 plus 8 is 46. 38 by 46 is 82.6%. So B is gone as well. What is sensitivity? True positives by true positives plus false negatives, which is 70 by 70 plus 5. 93.33. So, thank you, Shreya. All of you are right. Devangana, Kritika, Malini, Nagamani, Shreya, and I guess Purnima as well, right? So, six people, which is great. 8 plus 5. All right. Question 6. If A is a confusion matrix from a binary classification prediction, the value of A11 and A22 denote option A false positives and true negatives, option B true positives and false negatives, option C true positives and true negatives, and option D true positives and false negatives. Kritika, question 6, is it? Devangana says C. Okay, Kritika, we'll look into the answer. Just give it 1 minute and tell me what you think is the answer. So, you know what a confusion matrix is, right? So, previously, when we had Nagamani says D, that's nice. So, we have some difference in options. You have predicted on one side and experimental or actual on the other axis, and you have the distribution of true positives, true, sorry, false negatives, true negative, and false positive. Okay, so Malini goes with D and Nagamani has shifted to C. Ha, so that is the problem. So, confusion matrix is generally with, I don't have a snip of, if you remember the Vika, I don't have Vika installed in my system. So I cannot show it to you now like a pop-up window. But if you had seen this data displayed in the end, it was not in a box or matrix. It was just displayed in text, right? And there you had predicted or something, or calculator or something, and here you had the actual value of alpha, beta, and transmembrane uh, classification problem. Even otherwise, confusion matrix in general will have this organization: true positive, false positive, and true negative. But in one of the problems last week, was it last week or sorry? I guess some of you might have seen the answer. Mm, was it week 10 or 9? Okay, this one. This is the week. Huh. So in this question, it was given as actual on one side and predicted on the other side. But however it may be, maybe I should solve this now once and for all. Mm, Okay, it's the other way around, is it? As long as you understand that when you have something predicted as positive and actually it is negative, it's a false positive. And when something's predicted as negative and is actually positive, it is false negative. So, in either case, the values that are flipping are false positives and false negatives and not true positive and true negative. So, you should worry when the question is about. I'm sorry about the predicted experimental confusion here. Oh, I don't know why I can't choose eraser. Okay. Oh, I still can't choose eraser. Anyway, I mean it is still correct for this matrix. So if I had predicted on my left axis and actual on the other axis, so predicted positive, negative, actual positive, negative will be true positives, true negative, and here predicted as positive, actually negative will be false positive, predicted negative, actually positive will be false negative. So the only ones that are flipping are false positive and false negative here, right? Provided again you have positive and negative here. If you flip that, obviously uh, you will have true negative, true positive back here, correct? So if even if you're worried about the axis of where what is, if the question is about a11 and a22, you can still go with true positives and true negatives, which is an option C. Only on the sides, or only if you go in the same row, you will find true positive and false positive in the same, or true positive or false negative in the same row. Diagonally, it will be false with false and true with true. So they're calling it A11, A12, A21, and A22, where the first is the row and second is the column. Right? So first row, first element, second row, sorry, it's A11 only. So it is, if it were a matrix, this is the first row, first column element, and first row, second column will be A12. So the question is about A11 and A22, so it will be true positives and true negatives. If it were about false negative and false positive, maybe you should go with this form of classification, which is also there in the picture in week 11, where you have true positives, false negatives, and true negative. But it's always better to have the classification system on either side, because if my memory is right, even in Vika it was prediction on the x-axis and actual on y-axis, but I will have to go back and check that and paste again. I mean, as long as we have the concepts right, it should work. Just give me a second to check if my recording is fine. Okay. All right. So can we all agree on C? True positives and true negatives at A11 and A22. Okay. Thank you all. Okay. Question 7. In week, percentage split can be done for Only 66, only 33, only 50%, the any percentage. Thank you, Purnima. Others? Thank you, Vijayanta. Thank you, Malini and Devangana. So we're all clear on this, right? Any percentage can be entered to split the data set into training and test. So it's not just 66 or 33 or 50, it could be anything. Moving on to question 8. Vika is used for option A regression, B classification, C clustering, and D all of the above. Thank you all. Three votes for B. Great. Thank you, Karishma. So that's right. And Devangana. Great. I think this is the maximum response we have received. Happy. So Vika is used, yes, for regression and classification, also clustering. All of the above is the right answer. D. Output of Vika provides information on A. Sensitivity, B. Specificity, C. Accuracy, and D. All of the above. Thank you, Purnima. Thank you, Malini, Vijayanta, and Shreya. So, four votes for D. So, output of Vika, as we just by mistake or unintentionally discussed, provides information on sensitivity, specificity, as well as accuracy. You can derive those factors. Therefore, all of the above is the right answer. Question 10. In K means clustering, K is meant for A. Number of data, B. Number of features, C. Number of clusters, and D. All of the above. Thank you, Kritika. And Devangana. Thank you, Malini. And Vijayanta. Shreya, Purnima. So we are all clear that K means, in K means, is regarding the number of clusters which is given as an input when you, okay, Shreya sides with B, but it's not the number of features, Shreya. So when you have a huge data set that you want to cluster, let's say there are four different classes in it, or you want to classify it or cluster into three different groups, the three or four or six is the key. So the algorithm will try to classify or separate into so many clusters, and also improve the clusters, the members in each cluster, so that the mean within each cluster is minimal, right? So until the algorithm converges on overall differences means between each of those clusters, and internally between each of uh, the data or the centroids that are calculated for each cluster, which is the mean of the observations in that cluster, should be kept minimal. And for each of the observation you will calculate and assign it to the nearest cluster, or which is the closest group it belongs to, based on whatever features that you use as a parameter to calculate those means. So it's not the number of data or the number of features, but it is the number of clusters into which those data are going to be grouped based on some feature. All right. So option C is the right answer. So we move on to the current assignment, which had a last date probably 2 days ago, 2 or 3 days ago, Wednesday, I guess. So PDBTM provides the data on option A, structures of transmembrane helical proteins. Thank you, Purnima and Nagamani. B, structures of transmembrane strand proteins. C, structures of transmembrane helical and strand proteins. D, structures and sequences of transmembrane helical and strand proteins. And 3, 4, 5, 6 answers for D. This is a straightforward questions. Thank you, Ajayanta. That makes it 7. No confusion in that. PDBTM does provide structure as well as sequences of transmembrane, that's where the TM comes from, helical and strand proteins. Okay, question 12. Which machine learning algorithm is not based on supervised learning? A. Support vector machines. B. Naive Bayes classifier. C. K-means clustering. And D. Linear regression. Okay, 3 votes for C. Thank you, Bhavana. It also falls under supervised learning only. Although it wasn't discussed in detail, I believe. Linear regression, logistic regression, decision tree, SVM, naive Bayes, KNN were mentioned under learning algorithms, but under supervised learning, we looked only into neural network and support vector machine in briefly, not in great detail. And under supervised learning, K means clustering, principal component analysis, and self organizing maps were again mentioned. So, K means clustering does not fall under. Thank you, Malini and Nagamani Purnama. So, yeah, so K means clustering is not a supervised learning, rather an unsupervised learning algorithm. Option C is the right answer. Question 13. Choose an algorithm for prediction of protein stability, where in brackets you have real value. Seek to feature is actually, I have a small clip for it in the next page or slide. It is not a tool, not an algorithm for prediction of protein stability. You can input, it's a web server where you can input a sequence and get some of its features, but I don't know whether you can call it an algorithm for prediction of a real value for protein stability. Oh, so I've got 6 votes for linear regression, is it? Okay, I did not notice that. Which is right. So, for Kritika, Seek2 feature is a comprehensive web-based feature extraction tool which computes protein and DNA sequence-derived features. You can, um, personally, I haven't used that for. So this is a server developed by Dr. Bromley. So this is a article. Um, so you can see it's by the prof. It's a comprehensive web-based feature extraction tool. Machine learning techniques require which computes 252 proteins and 41 DNA sequence-based descriptors. And this is the interface. So although you have predict protein structures and expression profiles, the DNA sequence-based properties are physicochemical, enthalpy, entropy, and stuff associated with DNA and protein sequences, which can be useful in extracting valuable descriptors for prediction purposes. But you will not directly use this for calculating the real value of protein stability, right? Instead, again, similarly, you have principal component analysis and k-means clustering as learning algorithms which can help you predict something. But if you were to choose an algorithm for predicting a real value of protein stability with maximum, or the best way to do that would be to go for linear regression, because it'll be k-means clustering is an unsupervised learning algorithm. So is PCA. You would prefer, because and there's no means of, um, how do you say, evaluate the performance of these algorithms, whereas linear regression is much better in terms of reliability. Okay. Question 14. For a given method, sensitivity and specificity are 95% and 85.5%, 95.5 and 85.5, probably respectively. Okay. If we label the positive class as negative and negative class as positive, what would be the sensitivity and specificity? Okay. I've got 4 votes already for B. So if the classes are reversed, thank you, Malini. How will the sensitivity and specificity change? So if truly I have uh, 4 blue balls, 2 red balls, and okay, so true positives are 3, true negatives are is 1, and then I'm considering B as positive. Okay, and this is negative but positive, so false negative. This is false positive, correct? So my specific sensitivity is equal to positives by positive plus false negative. 3 by 4 is 75, and specificity will be number of negatives. Two negatives will be 1 divided by 1 plus um, false positive, correct? 1, which will be 15. Now if I were to change that R is positive and B is negative, the whole story changes in that TP becomes TN, TN becomes TP, FP becomes FN, and FN becomes FP. So therefore my sensitivity will become 1 divided by 2, and specificity will become 3 by 4. So it does flip, right? I mean, some of you might be immediately able to visualize this. For those who couldn't, it just flips. The namings flip over, and therefore sensitivity and specificity become interchanged. Okay. Question 15. For predicting the type of a test protein X between two groups of proteins A and B based on amino acid composition, which of the following statement holds good? A. Correlation between X and A is higher than B. It belongs to A. Deviation between X and A is higher than B. It belongs to A. Deviation between X and A is higher than B. And correlation between X and B is lower than A. It belongs to B. D, all of the above. Okay, 4 votes for A. Thank you, Shreya. And when you look into the options, right, correlation between X and A is higher, which means X is closer to A than B. So it belongs to A. Is that acceptable? Yes. Deviation between X and A is higher than B, which means X deviates. First of all, I don't know how what they mean by X deviates so much from A. Let's take it as opposite to correlation because it's not so correlated, therefore it is deviating from B, which means it's correlating better with B than A. So it cannot belong to A, right? This statement is false. Deviation between X and A is higher than B, which means X is closer to B and farther from A. Just spatially, I'm for visualization purpose. And correlation between X and B is lower than A. So these both are contradicting statements in each other. So if by statement 1, if B is closer to A than B, then correlation between X and B cannot be lower than X and A. Therefore, it cannot also belong to B. So this is no. If the second statement had not been there, C is an equally viable option, right? And since you already have two false options, not all of the above. We go with A. Question 16. In Vika, cross validation can be done for twofold, fivefold, tenfold, and all of the above. That's right. I see people typing in with great enthusiasm. Happy to see that. Can be done in all. I mean, I haven't seen anybody mention going above tenfold, but 2, 5, 4, 6 are also possible options. So all of the above is the right answer. Question 17. Weka accepts which of the following input formats? Dot ARFF format only, dot C only, dot great. 1, 2, 3, 1, 2, 3, 4, 5, 6. Dot JSON format only. All of these are acceptable, right? ARFF as well as CSV are accepted. JSON 2 is accepted because it's how JavaScripts are written, and we being a Java uh, program, it does accept. Question 18. What would be the output of the function f of x is equal to sine of w0 plus w1 into x1 plus w2 into x2? W0 is equal to 0, 1. Okay, Kritika, will solve now. I mean, I haven't solved this before, so we can do it together. Ta-da, ta-da. So, thank you, Vijayanta. If W0, W1, W2, X1 and X2 are 2, minus 2, minus 1, 0.5 and minus 1 respectively, let sine of x is equal to 1 if x is greater than 0 and 0 if x is less than 0. So if x is positive, is greater than 0, SGN of x is equal to 1. And if x is less than 0, SGN of x is equal to 0. And now we have W0 is equal to, thank you Shreya, 2, W1 is equal to minus 2, W2 is equal to minus 1, X1 is equal to zero point 0.5, X2 is equal to minus 1. So first let's do this equation. So those who are doing parallelly can finish off and give the answer. So W naught plus W1 x1 plus W2 x2 is equal to 2 plus minus 2 into zero point 0.5 plus minus 1 into minus 1. So this gives you 2 plus zero point 0.5 into minus 2 will be minus 1. 1 into 1 will be plus 1. So these both cancel and the answer is 2. Remember, you have SGN of this equation. So SGN of anything is equal to 1 if 2 is greater than or equal to 0, and 0 if 2 is less than 0. Which one is true? 2 is not less than 0, right? 2 is greater than or equal to 0. I mean, it is greater than 0. Therefore, SGN of 2 will be, or SGN of this equation is equal to 1. And if you see, even the options will not have 2. You should not solve this and immediately rush to the options. It is not 0, not 1. Oh, I'm sorry. It is 1. And I think I don't have an eraser. Oh god, I have it. Lucky us. Yeah. So when you see this equation and f of x is equal to sgn of some function or relation, you know that it cannot be zero point 0.5 and it cannot be minus 1 simply because it's not part of the options in the first place. Therefore, it has to be 0 or 1. Since it's positive value and greater than 0, it is 1. Was that clear, Kritika? Okay. So we can all agree on B and proceed. Question 19. Identification of co-expressing genes from mRNA expression data is an example of binary classification, non-linear regression, clustering, and none of the above. This exact example was mentioned in the lecture, but I did not take a snip of that. Thank you, Vijayanta Purnima and Shreya. Oh, Shreya says B. Thank you, Kritika and Malini. So, identification of co-expressing genes from mRNA expression data is not an example of binary classifications. That much all of us are clear. Therefore, none of these is wrong. Wait, not therefore. None of these is also not the right answer. It is more of a clustering problem or an example as I see it, and not a non-linear regression case. Because when you look into a gene pool and mRNA expression data, it is more of clustering which genes are co-expressed temporally, or if you want to find out which genes are present in which chromosome or which mRNA or product of which chromosomes or which genes are expressed together or one after the other. It's more of a clustering problem than a non-linear regression. However, I would like to add more solid uh, backing for this question as I did not have any snip of this data to go with. But clustering is the right answer. C is right. But for Shreya's sake, I would like to add something to the slides when I publish. Moving on to question 20. R square is a mathematical notation for A. Coefficient of determination, B. Pearson's coefficient of correlation, C. Spearman's coefficient of rank correlation, and D. Coefficient of variation. So I guess most of you already know the answer. Thank you, Jayanta, Kritika, Purnima, and Malini. That's right. We know Pearson's coefficient of correlation R, where you use it for positively or negatively correlated. Pearson's correlation coefficient is used to identify patterns in things, whereas coefficient of determination capital R square is used to identify the strength of a model. Capital R square is the coefficient of determination, and we know that it is used for determining the strength or identifying the strength of a model. Spearman's coefficient of rank correlation is this equation. This is Spearman's rank correlation coefficient. I haven't personally used it again. So di is the difference between two ranks of each observation. Okay, and 10 is the number of observations here. So I've just put this in here for extra information, and this is in continuation. Spearman's coefficient of rank correlation is a non-parametric measure of rank correlation, which is statistical dependence between ranking of two variables. It assesses how well the relationship between two variables can be described using a monotonic function. I did not include anything for coefficient of variation. Most that's more like covariance and stuff. So the right answer is of course A. Maybe I should erase uh, this simply because it is hard to erase afterwards. Okay. So here I've pasted the same question from previous week. We have already solved this. I wanted to change the equations too, but this was more in the last minute when I remembered about the calculator issue. So we'll use the same thing. I don't remember what the values of x, y, z were, but I guess we can always go back and find out, or one of you might remember. So the IC50 values of three protein ligand complexes are 6, 7, and 4 nanomolar, and IC50 is a function of hydrogen bond donors, hydrogen bond acceptors, and polar surface area. Okay, so we would have this as blah blah blah. Donors 2x, 3x, 2x plus 3y, 4y, 4y plus 1z, 2z, 3z. Okay, so we are going to solve these three equations without the aid of calculator. So let this be first, second, and third. So I'm going to multiply 1 into 2 so that you see, try to eliminate at least one variable, and you should do that with both. So if I'm going to eliminate z's here, then I should do that twice in order to get eliminate z's altogether and have just x and y. Then I can solve for two variables, right? Yes, I can directly do 1 minus c as well, because 2x and 2x are already ready made for me, and 4 and 4y is also easy, but I have to do x again. So I thought of starting with Z, but you can do how Vijayanta says as well. So if we do his way, let me erase, right? Okay. So 2x plus 3y plus z is equal to 6. And you also have 2x plus 4y plus 3z is equal to 4. If I subtract these, I'd have minus y minus 2z is equal to 2, right? Keep a close watch on what I solve because I could make mistakes. Next, you need to have another set of equations where you have eliminated x again. So I'm going to do, let's say, pick anything. Let's do 2 and 3 again. So first I'm going to multiply 2 with 3 and 3 with 2. Okay. So 6x plus 12y plus 3 2s are 6, right? 6z is equal to 7 3s are 21 and 2 into, oh, did I just okay. I'm sorry about the errors. And so that we have 2 into 3 6 and 3 into 2 6 to eliminate. So 3 2s are 6x plus 4 2s are 8y plus 2 2s are 4z is equal to 7 2s are 14 and 2 3s are 6x plus 4 3s are 12y plus 3 3s are 9z is equal to 4 3s are 12. Subtract again. You have minus 4y minus 5z is equal to 2. Okay, so I'm going to multiply 4 by 4. So you have minus 4y minus 8z is equal to 4 2s are 8 and you have minus 4y which I am taking here minus 5z is equal to 2. We are going to subtract again, so this is going to become plus and minus here. 4 plus 4 0 and you have minus 8 plus 5 is minus 3z is equal to 8 minus 2 will be 6. So z is equal to minus 2. So that much we have found, and if you substitute this into 4, minus y minus 2 into minus 2 is equal to 2, minus y plus 4 is equal to 2, minus y is equal to plus 2 minus 4 is equal to minus 2, minus and minus get cancelled, y is equal to 2. If you put this into any one of these equations, let's put this into 1, 2 into x plus 3y, 3 2s are 6 plus z will be 1 into minus 2 is equal to 6. So 2x, 6 minus 2 will be 4 is equal to 6, or 2x is equal to 6 minus 4 will be 2 x is equal to 1. I hope this was the answer we got last time as well. So you know x is equal to 1, y is equal to 2, z is equal to minus 2. Next question is the IC50 value of a compound with 3 hydrogen bond donors, 3x, plus 3 hydrogen bond acceptors, 3y, and a polar surface area of 2, z is equal to what? So I know it is 3 into 1 plus 3 into 2 plus 2 into minus 2, which is 3 plus 3 to 6 minus 4. 9 minus 4 will be 5. Was that the answer we got last time? Anybody remember? Thank you, Vijayendra. So the answer is right. We are done via. Oops. So you can see the same answers here, right? 1, 2, and minus 2. So it is possible. You just need to eliminate at least one variable and go ahead with solving the question. Was that helpful, Kritika? Yes. So this is one more question which we had done last week, but I had not, added any pictures to tell you why. So I thought you might find this useful. Dash generally happens when the gap between the training and test error is large. So is it good fitting, over fitting, under fitting, or all of the above? So we know it's not good fitting. I wanted you to grasp the difference between over fitting and under fitting better. This is optimal or good, whatever. So underfitting occurs when your model is too simple for your data. Overfitting occurs when the model is too complex for your data. So here is the test error and train error. When the gap between the test error and train error is huge, it is a case for overfitting. And when it is uh, too less, and when the model is too simple, when you have too little data to uh, and you're correlating too much, or you're predicting too much from your model, it is a case for underfitting. Okay, so I thought this might help. So that wraps up my question or the assignment that I have prepared for you all. It was good to have you all. I enjoyed being a part of this course and session. There's a doubt in a question like where we have to find hydrophobicity sequence. Thank you, Shreya. Which one, Kritika? Thank you all for joining today's session. Can you paste the question or tell me which week it belonged to? Amphithetic. Okay, so this was probably week 6 or 7. I don't remember. I'm checking my notes as well. I mean, if you know which week it is easier for us to go to that assignment. Okay, let's check 7 and 6. Thank you, Vijayanta. Final exam. Yeah, I believe it should help you to uh, score well if you go over the lectures and the assignments, all the sample questions of other TAs. I remember you had brought to my attention one of the issues with some live session. I had forwarded to the concerned person. I hope you got some redressal out of it. If not, I'm very sorry about it. And any suggestions? I really don't have any right now. I mean, just be calm, cool. Uh, go over the lectures and your notes probably and the assignments, and that should help you do well. So, Kritika, I don't. Oh, you mean this? I think I understand what you meant, Kritika. Okay, you mean the, yeah, this one, right? Something like this. Hydrophobicity values of these classes. I mean, it can't be too hard to remember, but I think, see, you have it given in the question here. So mostly it will be given. Okay. In case it is not, have a rough idea of what this is. Yeah. Generally, it should be given, but try to at least remember as to classify these as polar or hydrophobic. Have some acronym yourself to remember. I mean, these four are easier only. You just have five letters in each. You can make up some weird name to remember this. Generally, I hope it will be given in the question itself. Even if not, be prepared. Remember this part. Means some tiny small things like alanine, glycine. Actually, there's a power cut here, so I hope nothing stops. One second. Probably. I hope I'm still on. Do I have you all? I'm very sorry. There was a power cut, so I had to. Uh, oh, thank you very much. So I'm happy that you benefited out of this. But in case there is something, you can always mail me. I think most of you have my mail ID. So before the exam, if there's something I can help you with, I'll be glad to do so. And yes, okay. Thank you very much for being a part of this. And I learned and enjoyed learning new things with you all. All the best for your exams. And okay, so I'm going to close this session down and I will include this non-linear regression part to say it is indeed a clustering problem for that question, the mRNA clustering question, co-expressing genes. Otherwise, I guess we are done. So, all the best again. Do well. Thank you all and enjoy the weekend.